Okay, good afternoon, everybody, and I'd like to welcome Nathan Hubo, who is a third-year PhD student at the University of Southampton, and he's working on the role of jellyfish and their microbiomes on nutrient cycling and ecosystem productivity. And we're going to hear about some work that he did on the RGNO in 2019. So, Nathan, the floor is all yours.
Okay, I'm going to crack on directly with the introduction because I'm really short in time. This paper has been published in RGNO a few weeks ago. So the first thing I want you to know before starting with the experiment is that jellyfish are covered with complex microbiome. So a community of microorganisms that play a role in the fitness of the host, for example, participating in the innate immune system of the host, but they also participate in defining the ecological feature of their host. So then jellyfish and microbiome can be seen as one ecological unit. Second thing I wanted to know is that jellyfish, naturally, they excrete inorganic nutrients in the form of ammonium and phosphate. And when I started doing some trial experiment, I was really surprised to see a nitrate accumulation in my incubation. And that suggests nitrification. So nitrification is a usually two-step process, usually done by two different types of microorganisms. And that's an important process in the nitrogen cycle in the ocean. So the ammonia is first oxidized to nitrite, and then nitrite is oxidized to nitrate. So my hypothesis, so I designed my experiment to test the hypothesis that jellyfish are associated with nitrite and nitrate releases. And to test that, I've selected 4 different species of jellyfish. In yellow, you can see the purple sea nettle Chrysora fulgida that comes from the coast of Namibia. In green and blue, you have the compass jellyfish and the moon jellyfish that comes from the UK waters, coastal waters. And lastly, in red, you have the Japanese sea nettle that usually is around Japan, but in this case, it comes from the London aquarium. The reason is I wanted to try Medusa, a jellyfish that has been only grown in artificial conditions, so never in contact with natural seawater. So here on that map, you can see the distribution of these species. And on top of that, I've had added the squared area that shows the distribution of the cryptic species of the genus Aurelia. The moon jellyfish has many species, all cryptic, so same morphology. So then I expect that something that I observe in one of the cryptic species could be also applicable to the others. Okay, so now I'm going to explain the experiment quickly. When I catch a jellyfish, I first rinse it and then I incubate it for an hour in 2 liters of water. It's water that is artificial seawater, so bacteria-free. And the purpose of that incubation is to allow the mucus and the microorganism to be released in the water. After that hour, I will split that volume in 2. Half of the volume with the jellyfish will go to the jellyfish treatment, and then the other half with the mucus and microorganism, but without the jellyfish, go to the mucus treatment. The water is again bacteria-free, so artificial seawater. And in the mucus treatment, I simulate the presence of the jellyfish by adding ammonium in concentration that I expect the jellyfish to release. Of course, for each treatment, I have a control. And then I've been measuring nutrients every hour for 6 hours. And then I remove the jellyfish and continue measuring the nutrients for 2 hours. So right now, I'm just going to present the results of the jellyfish treatment because that's the only treatment where you have nitrate and nitrite accumulation. And actually, I'm going to skip this graph that shows the increase in the different nutrients. You can see that it's roughly linear. So that's why I'm going to present them as a rate. So here you can see in brown, the ammonium. So on the left, you have the rate that is so in nanomole per gram of wet weight. So it's normalized by the wet weight of the jellyfish and per hour. And as you can see, of course, ammonium is the main nutrient that is released. And remember, it's released by the jellyfish. But then you can see, and on the right, it's the proportion of these rates of the total nitrogen. You can see that nitrification can transform ammonium up to 1/3 of the ammonium released by the jellyfish. But also for some species, you have only, I don't know if you can see my pointer, my cursor, yeah. So here for the moon jellyfish, we can see there's only nitrate accumulation. And for the next to it, there's only nitrite. So if we have only nitrite, that suggests the presence of the ammonia oxidizer and the absence of the nitrite oxidizer. The nitrite is accumulating, but there's no nitrifiers to do the second step. No? Similarly, if we have only nitrate, then it suggests the presence of both nitrifiers, but then nitrite is directly oxidized to nitrate and doesn't accumulate. And when we have accumulation of both, then it suggests the presence of both nitrifiers, but the reactions are uncoupled, and that's why we see an accumulation of both nitrogen forms. So based on these results, we can suggest that nitrifying organisms are ubiquitous members of jellyfish microbiomes because we have 4 different species, even one that has never been in contact with natural condition, and all of them present some evidence of nitrification associated with them. And the other thing is the nitrifying community seems to be different with species. Have we seen? Sometimes we see evidence of one nitrifier, sometimes the other one, or both of them, but with different rates. So to put that quickly in context, when we have a jellyfish bloom, then we have a primary producer producing organic matter, and that organic matter will be stored into a gelatinous organic matter by jellyfish. And then that gelatinous matter is really rich in nitrogen, and some of that nitrogen will be released in the environment in the form of ammonium, and then further oxidized to supply then nitrite and nitrate. So then and these nutrients can be then reutilized by primary producers to stimulate primary production. So the jellyfish will supply a diversity of form of nitrogen in the environment. A last thing that we can do with my results is model the nitrogen release. So there's an allometric relationship between the weight of the jellyfish and their release. And thanks to that, I can model and come with this equation. And using this equation, then we can go to the measurement of density of jellyfish and see how much these blooms could release. And that's what I've done with these 2 cases. We can see on the left Japanese sea lake, and on the right the coast of Namibia. And these are measurement of density of jellyfish that have been done with sonar technology. So it's the best data that we have up to now. So now you can see the density of jellyfish, and I've applied my equations to transform that into nitrogen release. Sorry. So here you can see the transformation, and basically in the core of the jellyfish, in the core of the jellyfish, we have we can have more than 1 millimole of nitrogen released per square meter per hour. And now, if we want to see how relevant is that, we can compare it to the requirement of primary producers for primary production of these environments. And in the case of the sea lake, then that amount of nitrogen can supply up to 4 times the daily primary production requirement of that kind of ecosystem, and more than twice the daily primary production in the case of the northern Benguela ecosystem. Again, I want just to emphasize that these density are just a snapshot. So it's just in the core of the bloom that we reach these really high nitrogen releases. To summary, then, we have seen that nitrifiers are probably ubiquitous members of jellyfish microbiomes, and that these nitrifying communities are most likely to vary in composition, activity, and or abundance with the different species, and that jellyfish blooms can supply more than the total nitrogen requirement for primary production in the core of the bloom. And in the future, I would like to, of course, identify my nitrifiers to see if they are archaea or bacteria, to see where they are located on the jellyfish, And finally, to investigate the question of, are these nitrifiers species specific or environment specific? That's it. Thank you for your attention.
Thank you, Nathan. That's a lot packed into a short time.
Yeah,
Thank
I'm
you.
sorry.
And I think we'll move on to the next speaker now.
Okay, thanks.
Let me introduce Nancy. At this time, she's a PhD candidate in marine biochemistry and geology at Leibniz Center for Tropical Marine Research. She accumulates some 10 years working experience in aquatic research, working at Kemfree at Mombasa branch, and doing multidisciplinary research projects in this area. He's working with the examination of the sources and the quantification of fluxes of essential nutrients such as nitrogen, phosphorus, silicia, and the corresponding effects on phytoplankton community structure and the occurrence of harmful algal blooms. So she's going to tell us about the about the flux of nutrients in coastal waters of Mombasa in Kenya. So Nancy, please.
So I'm going to present my current work, which is basically a proposal of what I'm working with. I'm yet to embark on the main thing, my fieldwork starting starting August. So this is basically the background information of what I'm doing. It's based on the importance of nutrients NNP and the anthropogenic influence. So nutrient NNP limitations that is occurring in coastal waters in terms of its effect on primary production. I think much of it has been presented by many people in the previous work. And the basic requirement is the Redfield ratios, which is presumed to be 16:1, although this varies with the species of phytoplankton. But we have also the silicates that are required by specific silicoflagellates and diatoms for their crystal formation. So the supply of nitrogen and phosphorus varies a lot depending with the source. And this is the basis of my work. So due to limitation with increasing nitrogen and phosphorus in the coastal waters, it increases the primary production. So it increases the increase of nutrient and phosphorus. So this occurs at different variations, compositions because of the source, depending on the source. And this affects the upper organisms in the food chain, which end up affecting the whole ecosystem. Another problem we have is the supply of nitrogen components forms in different forms. We have the reduced form, which is the ammonia, and then we have the nitrogen forms. Most of the blooming species prefer the use of ammonia because it requires less energy in its utilization as a source of nitrogen. But we have the diatomic, which is important for fisheries production, uses nitrate, not only as a nitrogen force, but it is also used as a source energy stability in the marine environment. When we have a high supply of ammonia in the marine environment, it enhances the formation of the blooming species, which is dinoflagellates. And when we have low nitrogen, it will always facilitate the formation of cyanobacteria, which is a nitrogen fixing. And these are the blooming species with the production of toxins that leads to the formation of algal blooms, as I've presented here in the photos. So this ends up changing the whole marine ecosystem and affecting fisheries. I tried to present something a little bit here with what I did during my fellowship in Namibia in 2017. So we were looking at the macronutrients regulation of phytoplankton community structure, and we were able to look at the transect 18 and I don't know, GC was geochem, two transects. with my colleagues. So what I did, I looked at the nutrient concentrations, and I also looked at the ratios. Here I present only the N to P ratios. And as we can see here, the concentration, the ratios is below 16. This means that there is a high nitrate limitation in the waters in the Benguela. And then also you can see along in the southern part, the ratio is towards 0, meaning high phosphate concentrations. So this will lead to formation. There is a possibility of high cyanobacteria in this region because of lack of nitrates. And the only phytoplankton that can survive are those that can generate their own nutrients, so nitrates. So we also did the count of cells. We did some identification of the phytoplanktons or the herb species, but we also did the cell counts. And these were some of the results that we generated during that time, with high cell concentrations in the surface waters. And this was at the southern part. Here we also had very high nitrogen concentration, and we can see it's the place with the highest N to P ratios. So this means that towards the red field, when we have towards the red field, the environment really enhances production of the marine, of the coastal system, and this is very essential for fisheries. So back to what I'm currently doing, my current work that is based in Mombasa is focusing on the anthropogenic sources of nutrients. And I'm looking at the sources and the pathways and how they are going to influence the herbs formation in Mombasa. So my hypothesis is that increasing herbs development in Mombasa is as a result of nutrient inputs from agriculture, sewage, ballast water, industry, and tourism activities that are resulting from increasing coastal development. This is basically how my work is organized. I've split it into 5 different objectives, specific objectives, and I have 3 different stages. I'm going to look at the nutrient fluxes in terms of nutrient concentrations, nutrient ratios, the N forms, that is ammonia and nitrates. And then I'm going to look at the macroalgae, the carbon and nitrogen isotopes. So this will help me in establishing the sources. And I'm also going to look at herbs development in terms of primary production, biomass, species composition, bloom frequency and intensity. So this will be determined by because I'm going to do an 18-month field sampling for 18 months. And I'm also doing literature review on the current available work. And I will also use the satellite imageries for this. And the last part will be on coastal development, establishment of the changes in coastal development. This will be done later on outside, maybe outside part of my work. And it will be covering the land use, land cover changes that has taken place for about 3 decades. And I will also use the population changes in the area to see how sewage content has changed and the agriculture. So my specific objectives, I've summarized it together with the methodology that I'm going to apply. The first objective is to determine the spatial and temporal variation of nutrient fluxes, primary production, and herbs in the coastal waters of Mombasa. So this is what I'm currently doing, and I'm using secondary data based on available publications. And I'm also going to use the analysis of satellite images. And the second part is to establish the quantity and quality of nutrients associated with different sources and pathways. So I'm going to assess seawater, groundwater, pore water, and rainwater to see the different pathways and analysis of these. And then I have examination of primary production and herbs. So this is going to be done by microscopic analysis, and then establish the relationship, which will be based on data from 2 and 3. And then the last one through modeling. Thank you.
Okay, thank you very much, Nancy. And if you agree, so let's go to proceed to the next host and the next speech.
So Jana Pilatova is from the Charles University in Prague and Czech Republic, and in her 6th year as a PhD student. And she is an enthusiast for microalgae and protists, so small single cellular eukaryotes, and has been studying different aspects of their metabolism, like, for example, iron and copper and lipid production. But lately she has specialized more into analyzing the crystalline inclusions that they may have and what the nature of these inclusions is with Raman microscopy, a very strong recent tool. And one of her findings there was just recently published in Proceedings of the National Academy of Science. That is crystalline guanine inclusions, and she's going to tell us more about this now.
Thank you. Hello, everyone. I appreciate a lot to be invited to this forum. My name is Jana Pilatova, as Heidi said already. And recently, together with my colleagues, we have found microcrystalline guanine to be an unexpectedly high capacity and rapid turnover nitrogen reserve in microalgae. The nitrogen reserve of guanine crystals hasn't been studied in nature yet, and it hasn't been considered in nitrogen cycling either. And those are the things I see very interesting to be studied in the Benguela upwelling system. As you can see here, guanine crystals can be nicely observed in polarizing microscopy in different species of algae and all kinds of protists. And compared to well-studied organelles and cellular structures, the crystalline inclusions are still of unknown composition, functioning, and biogenesis. However, they have been observed since the very beginning of microscopy, being mentioned even by Charles Darwin and Ernst Haeckel, the greatest biologists of their era. Fortunately, nowadays, we have a powerful method for addressing the composition of cellular contents, the Raman microscopy, which is named after the discoverer of Raman scattering. It can be used in vivo and in situ. It's label-free and non-destructive tool. It's kind of a confocal microscopy connected with vibrational Raman spectroscopy, so that it's scanning through the cell and recording the spectral data in every pixel. And every chemical compound that's particular, has particular vibrational spectrum, a kind of a fingerprint or a barcode that's specific for different chemical bonds. And therefore, we can picture the composition of different cell inclusions. For example, the lipid droplets in a unicellular alga here, or polysaccharides or polyphosphates, etc. In a marine dinoflagellate of Amphidium carteri, together with plastids, polysaccharides, and lipids, we also found crystalline inclusions of guanine. Guanine is a nucleobase, a part of DNA and RNA. In form of crystals, it may have a different role though. Having 5 atoms of nitrogen, it acts as a high-capacity nitrogen reserve. We have used isotopically labeled forms of nitrogen to feed Amphidium, and it always produced guanine crystals quite fast, within, let's say, the first day. And then it started to disappear as the cell culture was growing, because the dividing cells use the nitrogen for producing proteins and nucleic acids. We also estimated that the maximum saturation level of nitrogen per cell might increase over 150 picograms of guanine per cell, which is just covering the consumption for cell divisions, for 3 consecutive cell divisions. So the culture grows 8 times from one cell. We also showed the guanine crystals to be important storage in symbiotic dinoflagellates, symbiodinaceae, in various species of corals, and also in other diverse marine, freshwater, terrestrial, and even acidophilic algae, with the majority of crystals being made of guanine, and one exception made of uric acid, which is also a kind of a purine. Recently, I have screened more than 200 species across the broad diversity of all eukaryotes, and I found that purine crystals are really universally present nitrogen storage, and compared to other biocrystals that I also found, such as calcite or oxalate, the purines are really prevailing species of biocrystals. In total, it was around 80% of all biocrystals I found. I've also conducted a first pilot study on seasonal dynamics of purine crystals in freshwater plankton here in Prague, in Vltava River. You can see it in blue. So the seasonal dynamic is really there, and it strongly correlates with the number of green algae, mostly the genus of Desmodesmus. And it negatively correlates with the presence of diatoms, and diatoms usually do not possess guanine crystals. There was only a weak correlation between the crystal presence and nitrogen or phosphorus, and no correlation with other nutrients in the eutrophic river waters. And now, in case of Benguela upwelling system, I would like to address at least some of the possible questions. What are the purine crystal containing species there? And then, more interestingly, are there correlations between crystal contents and nutrition availability in different parts of the upwelling system, and or even seasonal dynamics over time course? And last but not least, I would love to study the role of purine crystals in nitrogen metabolism and biogeochemical cycles. So thank you for your attention, and I'm very looking forward to the discussion later on.
Because Kathleen was one of our students in the very first RGNO course in 2014, together with Elizabeth Julies at the time. Now, her resume is a continuous advance through learning and accepting challenges, I would like to say, because she started as a research technician, then became a teacher at an agricultural and nature conservation school, then was appointed as a lecturer at the Windhoek College of Education. Then she became an assistant in marine and desert ecology, then a lecturer at the Faculty of Science Department in Biological Sciences and teaching aquatic ecology and conservation. And finally, she has an MSc in biodiversity and management from the Humboldt University in Berlin. And this qualified her beautifully to become now a microbial ecologist of marine systems. Her talk will be potential ecological impact of an invader mussel, Semimytilus algosus, on native mussels along the central coast of Namibia. Kathleen, are you ready?
Okay, thank you so much. My talk today will be on a much lighter note, as I'm a teacher, and I'm going to talk about work that I've done over the few years. I started in 2014 at the university, so I've really done a lot of work. So my title is Potential Ecological Impact of Invader, that is Semimytilus algosus, and what impact it has on the native mussels along the Central Coast. Now, this presentation was also assisted by my intern, Angelique Dodds. Good. My keywords, I'm not going to focus on that. But first of all, I would like to speak about capacity building, because I'm research project coordinator at a university. And over the years, for example, I supervise, I counted today 22 students um, that did topics related to the Rocky Shore intertidal zone. And that means they will go out for a weekend, 5 days. Sometimes we go out for twice a year, and then they collect data on the coastal areas, especially the intertidal zone. So this data that I'm going to present today was collected throughout the years, and we started in 2014. This is just to show you my students and various occasions when we visited the intertidal shore. I also would like at this stage to mention Dr. Julies, because she was really instrumental to spark my interest in marine ecology. Now, first of all, I would like to say that semi-mytilus is invasive mussel, and it belongs to the class Belvades. And in Namibia, we focus mainly on 4 different types of mussels. We've got the 2 native mussels, that is the perna and the corymytilus. Maybe I must also just get my my pointer, then this will be much easier. Good. This is the perna. Perna is the brown mussel that's quite clear. We can distinguish it very clearly if you do your collections. This is another native mussel, the corymytilus marialis. Sometimes this is very difficult, especially if it's very small, if it's still in the juvenile phase or recruit, to actually distinguish this from mytilus. But as you get used to the different types of mussels, you can actually clearly distinguish the different mussels. There are 2 invasive mussels. There's mytilus, that we're all familiar with, as well as semi-mytilus, that is the small, the bisexual mussel. Now, I'm not going to speak about mytilus today. I'm just going to focus on semi-mytilus. Now, what's very important is that these mussels, they spawn. So that means if they spawn, then the villages or the life cycle, they start, there's larvae, they will drift. Sometimes they will drift for kilometers before they find the space to settle. So they need like a substrate to settle so that they can start with their recruitment patterns. And what I've learned or read is that semi-mytilus recruitment will take place all around the year. So that means they've got a very high productive recruitment rates. Okay, this is just the life cycle I wanted to explain that I already said. So they spawn, and then they they form a valinger, and then they will just now drift until they find a suitable place to settle. Good. Now, I'm very interested in the history of this species, and a lot of the research already has been done on this topic, on the invasion history. And what's very important, or not important, but interesting to me, is that this semi-mytilus is native to the west coast of South America, from Ecuador to Chile. And what happens is that this was introduced in the late 1920s already in Namibia. And that was recorded by Mead et al. In 2010. And it also looks like this invasive species, that is semi-mytilus, was then introduced from Namibia to South Africa. So that's also just to note. Now, what's important is that I would like to show you is that if you look at the distribution pattern of the invasion pattern of this mussel, then you will see that in South Africa, it's really already well established. And in Namibia, we found that they found in Long Beach, Langstrand, Cape Cross, and also Terrace Bay. So it's quite, it occurs quite frequently in Namibia. Now, where did they get all this data from? That is by monitoring the rocky shores. So we have researchers in South Africa, like Tammy Robinson, Skyn, Zeeman. They will come and they will on a monthly basis actually go out to the rocky shores, and they will then note and count and monitor the rocky shores. So that's how they compiled now this history of the species. Now, I'm not going to say much about the morphology because I don't have a lot of time, But very important, you can see it's a very small muscle. It's less than 40 millimeters, and it's very easy to actually to distinguish if you're on the in the tidal zone, because it's smooth, it's elongated, it's green, it's dark, it has like a bulge below the apex. And if you want to make sure really if it's semi-metallous, you need to open the muscle to make sure that you can actually see all the features. There's a real ridge over here, and also there's no pits in the, while the other muscles you will find there's pits in the residual ridge. What's important to note is that this muscle is bisexual. That means there's both gonads, the male and female gonads. Good. Now I included these three graphs because I want to show you that how this population, how this semi-metallus spread. Now this is the 2017 data. As you can see here, I've got semi-metallus over here, then I've got penna. Corymetallus already was also well established, or is well established. And as you can see, 2018, there was a huge increase in the abundance of semi-metallus, and also in 2020. Now I'm busy now, we're still busy with to analyze 2021's data, and I didn't have the chance to do that. So as you can see here, it's actually well established. And if you look at Corymetallus, we'll see they almost close the type of the numbers, but that is the data that I wanted to present. Good. Now the next graphs or figures is the ordination diagrams, and that is based on the number of on population structure. So now I look at the juveniles, recruits, the adults of the three different species. This is now semi-metallus, this is Corymetallus, and this is now the penna. And as you will see here, that all these, both this species, they dominate the lower shore. So you'll find them mostly in the lower shore, while especially in wave exposed areas. And especially semi-metallus is very intolerant to desiccation. So that means you won't find semi-metallus higher up there in the tidal zone. So you'll find them subtidal and in the lower shore, and that is also where you'll find Corymetallus as well as penna. So that's why I wanted to look at the impact, because this muscle, this invasive muscle, is not like metallus. Metallus is more mid-zone dominated, and so that means it can actually coexist with penna, but not but can't survive in the lower zone. So what I found is that the adults, as you can see here, of semi-metallus are more common in the lower watermark, in the low zone, and the juveniles and the recruits, they are evenly distributed. While in the case of Corymetallus, you will find that the juveniles, they are mainly now more at the mid zone, while the adults and the recruits you will find more in the lower zone. And in the case of penna, the adults, they are distributed between the low watermark and the mid watermark, and the recruits, you will find them more in the low watermark. So I just wanted to show you that that all these three muscles, they really they're all in the lower zone. Now my interest was now, because we've got all this data, so we ask each other now, the student, so what impact does semi-metallus have on the native muscles? And so what we've done, we did a simple linear regression, and from 2018 as well as 2020. And what we found is there's a significant positive impact of semi-metallus on Corymetallus. And you can see the regression value is 0.2, so it's about 20% that you've got this value. So that means it's a significant positive impact. So as semi-metallus numbers grows, it also the numbers of Corymetallus will also then impacted positively, and that's the same with with this penna. So the regression here was 0.17, so it's also very positive impact. It is also significant impact on the numbers of penna. Now in 2020 we have the same. As you can see here, once again, look at the impact. Again, very significant, positive. So our question now is, so it's such a positive impact that these semi-Mytilus has. It's an invasive species, but it has a positive impact. And if you think about this invasive species, it's only about less than 40 millimeters in size, in length. And that length suits a variety of predation, or let me say predation types. And we know, for example, that these mussels, they are really forage species. So what's going to happen? So what we find is that because they dominate open rocky shores, that means their recruitment rates are very high. They will really produce at high rates. And this might ecologically impact structural complexity of the rocky shore structures, community structures, you name it. But what's positive is that this will form like an abundant prey resource, because as we know that many of our predators, they forage on mussels, they consume mussels, like the whelks, the rock lobsters, the starfish, the octopus, and you name it. So they feed on this abundant prey, and this prey falls into a size range that actually fits most of the predators' preferences. So, but what I also read is that there are a tendency that some of these native predators, they still feed on on a native species. So some of them, for example, like the whelks, they incorporated alien mussels in their diet, but not the rock lobster. And why not? So these are still questions that we would like to answer, and would like to seek assistance and to investigate and to run experiments to see why is it like this, and why, for example, another question, for example, what if this native predators feeds on native mussels, then they will also facilitate the invasion of this mussels, because they remove now the native competitors. So that means they will also facilitate now invasion of this mussels. So that is really what you would like to look at, the potential impacts, but ecologically as well as now, and see what impact it will have on our shores. Well, so I would like to conclude that I realize there are still extensive research we need to be done in Namibia, but what is a positive thing is that this relationship, or this impact, is positive. So we can look into predator-prey relationships, and also look maybe at economic impacts, and look if there's perhaps also negative impacts regarding this invasion. All right. Thank you so much. That's all from me. This is my key references. Thank you, Kurt.
Good morning, good afternoon, everybody. I'm going to introduce you Damona Henda Mateus, shortly known as Damona. She's an MSc candidate working on benthic invertebrates, and she's doing her MSc with the University of Namibia in Henties Bay. She participated in the 2019 RGNO camp. Recently, she joined the Namibia Nature Foundation as a seabed bycatch mitigation support officer with the Albatros Task Force in Walvis Bay, where she is mainly tasked with gathering seabed bycatch data. Furthermore, she plans to pursue her PhD study in marine benthic invertebrate research. She seems to be so passionate about. So yeah, in short, that is her. Damona, we are live. You can take it away.
Thank you, Dr. Kainge, for introducing me. My name is Damona, and as I was introduced by Dr. Kainge, I'll give you a brief presentation on my work for my master's study on spatial distribution and diversity of soft bottom benthic invertebrates along the Namibian coast which I did under the supervision of Dr. Kainge and Dr. Mafila. So basic invertebrates are organisms that lack a backbone, and they are normally found at the bottom of the water body, or some species they burrow in the sediments. And groups such as polychaetes, mollusks, and crustaceans are some of the marine benthic invertebrates, and we were looking at the organisms that are less mobile or sessile organisms. So their biodiversity is known to be long-lived, slow-growing, and also very slow to recruit when they are faced with sustained fishing pressure. These organisms are known to be affected by demersal trawling. And demersal trawls are large, heavyweight nets that are pulled through the water body with the bottom edge of the net or parts of the net of the trawls touching the seabed. And they are equipments that are normally used to target the bottom-dwelling species, and they are known to be very highly non-selective and can accumulate and destroy huge amounts of non-target species, including the benthic invertebrates, and can also cause extensive destructive effects on the seafloor communities. So on this slide, it's just showing, according to literature, some trawling activity are known to have a direct effect on the fishing assemblage structure, on the benthic assemblage structure, and also on the species or sediment composition. And these structures are also having indirect effects from the hydrographic variabilities. So the objective of my study was mainly to assess the distribution of and the diversity of the benthic invertebrates from the demersal trawls along the Namibian coast, which is collected as bycatch opportunistically during the Ministry of Fisheries and Marine Resource annual biomass surveys of the monkfish, the hake, and also the deep sea red crab surveys. I collected my data since 2018 to 2020, and the maps are just indicating the stations from where I collected my data. From the monk survey I collected in 2018, and from the hake survey I collected in 2 different surveys in 2019 and in 2020. And the crab surveys are only done in the northern part of the Namibian coast, and I did the surveys in 2019. So the monk surveys, they use the standard commercial albatross bottom trawl, which is a beam type of trawl. And while the hake surveys and the crab surveys, they use the Kisan Super 2-panel otter trawl. And that is the main gear that is used in the commercial fishing vessels in Namibia. So the stations were mainly collected from depths between 100 to 800 meters. So we collected data at every trawl station on board, and when the trawl is on deck, we account for all the benthic invertebrates, and sometimes when the trawl is so large, we collect a subsample. So when we are collecting a subsample, we collect per the Ministry of Fisheries sampling protocol, and we sort out all the invertebrates identified to their lowest possible taxonomic level, count and weigh, and also record using the rising factor in order to account for the entire catch. So we also take pictures of all the specimens, and for the specimens that we are unable to identify on board, we have to preserve them in 96% ethanol for further identification on shore. So from the 3 surveys, I analyzed a total of 324 trawls, and I accounted about 145 species belonging to 17 taxa. And from the monk survey, I recorded 55 species and 116 species from the hake survey and about 38 species from the crab surveys. So the maps here are just indicating the distribution of species that I have accounted most often from the surveys, and it's showing the Assyrian communities, which was normally found in the central region of the Namibian coast, and not much was found in the far south of the coastal of Namibia, and far in the north, we didn't account for much of the Assyrians, and they were only found between the depths of 100 meters to 400. The Sybarites community are the sponges or the poriferous. We accounted higher abundances almost along the entire coast, and they were found between the depths of 100 to 700 meters. There was also Procesta penicillatus, which is a starfish. Not much of the abundances, but they were only found in the far south of the Namibian coast, and they were found between the depths of 100 to 600. The crabs, Bathinectes piperites, they were found between the depths of 100 to 800 meters, and they were having a fairly similar distribution along the entire coast. Also, the squilla and the Brysopsis lyrifera capensis, which is a sea urchin, also not much of the abundances, but they were found mostly from the central region towards the southern region. So diversity of the benthic invertebrates was assessed per latitude or per region, and also per depth and temperature. And the results are showing that there was a significant difference in species diversity along the Namibian coast. From the Hague survey and the Monk survey, it's indicating a statistically significant difference in the species diversity, except for the crab survey, which was mainly based on the northern region only. It didn't show any much of the difference in the species diversity. And species diversity across different bottom depths, also it shows not much of the statistically significant difference in the species diversity. So the mean species distribution is quite similar, although it varies. It didn't show any statistically different distribution. And the species diversity across different temperature ranges, it showed a significant difference in temperature distribution of the species from the crab survey and from the Hague survey. Except for the Monk survey, it just showed similar distribution of benthic invertebrates at different temperatures. So mainly the distribution is showing that at lower temperatures, there was higher distribution, or there was a higher diversity of species at lower temperature. Like as you see in the graphs there, it's showing a higher mean species diversity at the lower temperatures compared to high temperatures. So although this data was based on a single survey, it covered a larger scale area and provided enough data to make accurate conclusions that with regards to the benthic invertebrates along the Namibian coast, and the abundances and biomass of the species was high, and their distribution varied along the coast. So more studies are needed on the identification, like the genetic analysis, and also the influence on environmental factors. And therefore, this study provides enough knowledge on benthic invertebrates distribution, and it can further be expanded to study more on the invertebrates, such as the habitat mapping. So these are just some of the species that I have encountered during the data collection. And thank you for